In this video, we will talk about the splicing and rules. This is genomic DNA having exon 1, exon 2, and exon 3. It also has intron 1 and intron 2. As we have discussed that, the order of exons in a gene remains the same as the order of exons in the processed messenger RNA. So, order remain in the same order in messenger RNA as in DNA, but distances along the gene do not correspond to distances along the messenger RNA or polypeptide products. Let's understand it. The distance from A to B in the gene is smaller than the distance from B to C, but the distance from A to B in the messenger RNA and polypeptide is greater than the distance from B to C. So we can say the length of a gene is defined by the length of the primary messenger RNA transcript instead of the length of the mature messenger RNA. All exons of a gene are on one RNA molecule, and their splicing together is an intramolecular reaction as exon 1 and exon 2 from same RNA molecule splicing together. Similarly, exon 3 and exon 4 from same RNA molecule splicing together. Both are intramolecular reactions. There is usually no joining of exons carried by different RNA molecules. So, there is rarely cross-splicing of sequences. Consider 2 RNAs molecules in cis-splicing. Exon 1 joins exon 2, while exon 3 joins exon 4. But in trans-splicing, exon 1 from one RNA molecule joins exon 4 from other RNA molecule. Similarly, exon 3 from one RNA molecule joins exon 2 from other RNA molecule. So we can say, in a process known as trans-splicing, sequences from different messenger RNAs are ligated together into a single molecule for translation. Now let's discuss the mutations in exon and intron. Well, mutations that directly affect the sequence of a polypeptide must occur in exons. But what are the effects of mutations in the introns? Well, the introns are not part of the mature messenger RNA. So, mutations in them cannot directly affect the polypeptide sequence. However, they can affect the processing of the messenger RNA production by inhibiting the splicing of exons that cause an intron to be included. They might cause an exon to be left out of the product. Intron mutations can affect not only the production of a polypeptide, but also its sequence. About 15% of the point mutations that cause human diseases disrupt splicing. Now let's discuss. Exon and intron base compositions differ using 4 rules. The 4 rules for DNA base composition are the first parity rules and second parity rules, both also known as Chargaff's rules, the cluster rule, and the guanine cytosine rule. Exons and introns can be distinguished on the basis of all rules except the first. Now let's discuss first parity rule for duplex DNA. In the 1940s, Erwin Chargaff initiated studies of DNA, base composition. That led to 4 rules. Beginning with the first parity rule for duplex DNA. This rule applies to most regions of DNA, including both exons and introns. Base adenine in one strand of the duplex is matched by a complementary base, thymine in the other strand, and base guanine in one strand of the duplex is matched by a complementary base, cytosine, in the other strand. The bases form weak hydrogen bond. By extension, the rule applies not only to single bases, but also to dinucleotides, trinucleotides, and oligonucleotides. Thus, guanine-thymine pairs, with its reverse complement, adenine-cytosine, and adenine-thymine-guanine pairs with its reverse complement, cytosine-adenine-thymine. In addition to the well-known first parity rule, later work by Chargaff led him to propose a second parity rule. The little-known second parity rule is that, to a close approximation, there are equal amounts of adenine and thymine, and equal amounts of guanine and cytosine in each single strand of the duplex, like the first parity rule. This extends to oligonucleotide sequences. For example, in a very long strand, there are approximately equal numbers of adenine, cytosine, and thymine, guanine dinucleotides. The reasons for the existence of this rule are not clear, but sequencing of many genomes has shown it to be nearly universally true. The second parity rule applies more closely to introns than to exons. We can say, if there are 6 thymines, 1, 2, 3, 4, 5, and 6, there will 6 adenines, 1, 2, 3, 4, 5, and 6. If there are 6 cytosines, 1, 2, 3, 4, 5, and 6, there will 6 guanine, 1, 2, 3, 4, 5, and 6. Moving to cluster rule. Purine includes adenine and guanine, while pyrimidine include cytosine and thymine. This cluster rule, as applied to exons, is that the purines, adenine and guanine, tended to be clustered in one DNA strand of the DNA duplex, and these are complemented by clusters of the pyrimidines, thymine and cytosine. The fact that in single-stranded DNA, an oligonucleotide is accompanied in series by equal quantities of its reverse complementary oligonucleotide. Finally, there is the guanine-cytosine rule, which is that the overall proportion of guanine-cytosine in a genome, or guanine-cytosine content, tends to be a species-specific character, although individual genes within that genome tend to have distinctive values. The guanine-cytosine content tends to be greater in exons than in introns. For example, human genome has 40% of cytosine and guanine and 60% of thymine and adenine. But yeast genome consists of 38% of cytosine and guanine, and 62% of thymine and adenine. Chargaff's 4 rules are seen to relate to characters, or pressures, that are intrinsic to the genome, contributing to what was termed the genome phenotype. So this was all. In our next video, we will discuss about the comparison of exons and introns of different species.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.